So as Bridget said, I'm Dilara, and I'm a former OmicsLogic research fellow, and I'll be taking you through the process of conducting research on astrocytes using using omics through the T-bioinfo platform. First, I will tackle some key points in research, like how to come up with a hypothesis, where and how to find good data, and how to convince an audience that your research is relevant and important. Then I will take you through my research and show which data and graphs are best to present and which ones I chose. And we will even practice a pipeline practically. So the hypothesis could be the start of your research or vice versa. For me, for the purposes of my research, I decided to start here. So while I was reading articles to find a topic that intrigued me, I came upon the gut-brain axis, and since my preferred field was astrobiology, I decided to study how the gut microbiome affects the nervous system of astronauts. My hypothesis was based on how high LET, linear energy transfer, ionizing radiation affected the nervous system of astronauts. My hypothesis was based on this type of radiation for several reasons, like the fact that it causes chemical reactions. I surmised that this type of radiation would have adverse effects on the human body and the gut microbiome. Now, on to finding a good data set. You can actually search for articles that are published in high impact factor and well-known well-known journals, because they are very well peer-reviewed, very rigorously peer-reviewed, and you should also try to pick ones with high sample numbers, since you can get better results in machine learning techniques with huge numbers of samples. As you can see, mine was categorized based on the radiation and time, and I had 80 samples. And of course, you can search NCBI and some other platforms like JGI for microbiome data. And as for the pipeline, it is important to choose an appropriate pre-processing and processing method, since you have a lot of data that you have to filter through when you have huge amounts of data. And you can do this using the T-bioinfo server. So if you would like, we can try a hands-on example, and then I can take you through which graphs I chose from those results. And then we can continue like that. So I guess the link is already in the chat. So if you could quickly go there, and you will come to this page where there are numerous different areas of analysis. And from my type of data, I chose Amplicon 16S, 18S because of the type of data that was in the article was 16S rRNA data. So here you can see a demo. And if you are following with me, we can, here you can see as well, pre-processing. For this demo, there isn't pre-processing, but for my data, there was, which helps remove primers, etc. And here we are trying all of them. I hope everyone can see, because I can't see the chat right now. So if you have any problems, please let me know. And what is great is the Data2 pipeline here and the phyloseq pipeline, you can run them both together at the same time. And then here you can see, I will pick this one, the Data2 and phyloseq plots PDF. And if you go there, here, you can see the abundance plot here, a heat map showing the differences in the abundances, a phylogenetic tree showing both the abundances as well here, the Shannon index that I will be explaining in detail. And here we have NMDS plots So you can go through those in your free time if you would like. And now I'll go back to my data specifically and show you. So basically, my NGS data, which is the 16S rRNA data, was collected from feces samples of mice exposed to different types, to 4 different doses of radiation. And these samples were collected at 10 days and 30 days post-radiation. So from these results, I decided to pick the NMDS plot, which is a non-metric multidimensional analysis plot, which is a data reduction technique, which means that you can input high amounts of data and it will, the machine learning algorithm will filter through this data and it will only give you the points which have great variance and have a great effect on the data. So you can see here the differences between the groups, and at 10 days post-radiation, that group is slightly more separated than the others. So the next output that I chose was the Shannon index. Since it gives us a general idea about the richness and evenness of bacteria in the samples, it is a great plot because it summarizes everything, the alpha diversity, the beta diversity of the microbiome. And here you can see that for lower doses of radiation, like 0.1 and 0.25 GY, the diversity decreases more compared to the control group. So next, I decided to use the phylogenetic tree, and this one has a bonus because we can see the abundances based on the categories as well. As you can see, the OTUs that have a great effect as here, the algorithm filtered those out for us. So OTU 3, 4, and 5 are the ones that I'll be focusing on in the next step. And here you can also see that for 0.1 GY at 10 days, it has the greatest decrease in abundance compared to 0 GY. To corroborate the results, I also conducted a t-test and an f-test on Excel. And using the tBioInfo server again, in the utility section, you can even generate heat maps. And that's why it's really great using this platform, because I didn't know coding very well, and I can't say I'm very efficient at it yet. So you don't need to know these things. You can directly start generating graphs without knowing how to code using this platform. So I generated this heat map, and as you can see, all the graphs that I use are the graphs that summarize large sums of data, which is helpful when explaining a new concept to an audience. Here you can see the difference in abundances for OTU 3, 4, 5, and 8. They are significantly decreased between 0.1 and 0 GY. I then decided to search the KEGG database for these 4 OTUs because I didn't have names exactly. So and they corresponded to Lactobacillus and Bifidobacterium genus, and I found out that these have protein coding genes in the GABA pathway. GABA is gamma-aminobutyric acid, and it is a chief neurotransmitter in the central nervous system and causes depression and anxiety when in low doses. So we can conclude that at lower doses of radiation, especially 0.1 GY collected at 10 days post-radiation, the abundances of these 4 types of bacteria decrease the most. And since they produce GABA, we can say that it has a correlation to the depression and anxiety that astronauts suffer during long-term flights. And that my initial hypothesis about the gut-brain axis was also correct. Of course, further research is required, but this is a good precursor study, and probiotics can help alleviate some of the symptoms of these astronauts. So what did I learn personally during this project submission and while conducting this research? First of all, I learned how to sort through good data and find good quality articles. And you become more efficient at creating a narrative so that your readers and listeners can understand your research. The more you write, the better you get at it. And especially referring to your mentors during this stage is great, because they will help you throughout the process, and you can efficiently estimate the quality of the data as well. And then you will come out also knowing which graphs are best to recapitulate your data and how to summarize your research. And overall, if you are rigorous during this stage, you can also learn how to conduct scientific data in an excellent manner. So here are my references, and if you have any questions, feel free to ask me. You can inquire anytime. And thank you for listening.